Hello everybody, thank you for watching the video about our deep learning-based method, DeepScena, for hierarchically detecting rare cell populations from scRNA-seq datasets. I am Ryu, a junior student from New Jersey. This video will mainly cover three parts. 1. The overview and discussion of deep learning techniques used in DeepScena. 2. How to download, install, and run DeepScena from GitHub. And 3. How to interpret your results. So first, let us look at an overview of DeepScena and why we designed DeepScena. Single-cell RNA sequencing is a widely used technique for characterizing individual cells and studying gene expression at the single-cell level. Clustering plays a vital role in grouping similar cells together for various downstream analysis. However, the high sparsity and dimensionality of large scRNA-seq datasets pose challenges to clustering performance. Although several deep learning-based clustering algorithms have been proposed, most existing methods have limitations in capturing the precise distribution types of data or fully utilizing the relationships between cells, leaving room for improvement in cell clustering performance, particularly in detecting rare cell populations from large scRNA-seq datasets. Rare cell types are specific cell populations within a tissue or organism present in very low frequencies, but still play a critical role in specific biological processes. In this figure, figure B, you can see the process of which single-cell RNA sequencing is used as the data for clustering cells. So we take the raw data from single-cell RNA sequencing in the form of a cell-by-gene matrix, and we cluster the cells based on that data. So in light of this, we propose that the strategic application of hierarchical clustering involving the definition of distinct features for different clustering levels could potentially facilitate the efficient detection of rare cell types. Let's look at the design of DeepScena. We designed an enhanced deep learning method called DeepScena for cell clustering by fully equipping the features of data imputation, dimensionality reduction, efficient clustering strategies using pairwise cell similarities, and hierarchical clustering for detecting rare cell types. DeepScena employs a NB negative binomial autoencoder to accomplish data imputation and improve accuracy. This is shown as Module 1. It then uses deep clustering with self-supervisation model that can gradually update pairwise data similarities during training. This is shown in Module 2, also called MNet. To perform hierarchical clustering, DeepScena recalculates the featured genes for each level in a top-down procedure. Let's look at the performance of DeepScena. DeepScena was tested on 8 large scRNA-seq datasets, and the results show that it can not only recall popular cell types, but also delineate novel rare cell types. So in Figure 2, this is the performance and comparison of DeepScena. Specifically in Figures C and D, this is the comparison of ARI and NMI scores, which are indexed to measure how well cell clustering is and how accurate it is, with higher numbers being more accurate. DeepScena is the first plot in every graph, and overall, across all datasets, DeepScena is superior to the other techniques. Figure 3 shows the performance of 6 tools on different dropout rates. High dropout rates means a large amount of genes that appear to have zero or low expressions in a single cell, and this is usually due to technical limitations in single-cell RNA sequencing itself. Second, let's discuss how to download and install DeepScena. DeepScena is publicly available on my GitHub website, and you can directly download the whole package by clicking Code. You can access my GitHub website by simply searching Sophia Chen Ko's DeepScena or going to this link. Once you're at my website, you can click Code and download zip to download the zip file of DeepScena. Please note that the code of DeepScena runs with Python 3.9 and requires PyTorch 1.17. You should also have CUDA installed. Please create a PyTorch environment, install PyTorch, and some other packages such as numpty, pandas, scikit-learn, scampi, and coda coolkit. See the requirements.txt and install.txt file for an overview of the packages in the environment we use to produce our results. If you are not familiar with different scRNA-seq data structures, we have prepared scripts to help you prepare input data from your own raw data, or you can download it from the NCBI-GEO database. For data preprocessing, please run the following command line to preprocess the UMI count data. Or you can also use this line. This program accepts txt, CSV, and 10x_mtx files as input file names. If the scRNA-seq data set is read count, please add the parameter -r or --reads as follows. You can also command Python preprocess_data.py-h for usage help. Once your data is ready, you can train the model and cluster your cells by using the command line python run deepscena.py. Before running DeepScena, please modify your file path and names. Two files, the preprocessed scRNA-seq dataset file and its cell type file. Rename your data set name, set the number of clusters, and reset some other parameters such as batch size and maximum iterations. The latent low dimensional space will be saved as dataset_name plus _usespace.csv, which can be used for visualization purposes. The clustering file will be saved as dataset_name plus _clusters.csv. Once you have successfully installed and prepared your data, you can run DeepScena for your data analysis. Based on different dataset size of cell numbers, it may take some time before you see your outputs. Now, let us explore two real case studies in cancer and computational neuroscience to demonstrate how to professionally discover rare cell types. Case study number 1. DeepScena was applied iteratively in a top-down process to detect rare cell types using the prefrontal cortex PFC cells from 12 mice, totaling 24,822 PFC cells merged from saline and cocaine conditioned groups. Initially, DeepScena detected 10 clusters, recovering major cell types identified in the original analysis using the SURAT pipeline. Subsequent analysis predicted top 25 differently expressed genes for each cell type, revealing cell-specific expression profiles. Notably, DeepScena delineated additional neuron types among excitatory neurons clustered together in the initial analysis. Further iterations revealed 8 subpopulations within these neurons, including layer 2-3 pyramidal neurons, layer 4 spiny stellate neurons, layer 5 pyramidal neurons, and layer 6 pyramidal neurons. Differently expressed genes, DEG analysis confirmed the specificity of certain genes to particular layers or cell types. Clustering results of the second iteration demonstrated DeepScena's ability to reselect feature gene sets effectively. However, subsequent iterations showed that highly variable genes did not significantly differentiate 7 clusters of the layer 2-3 pyramidal neurons. Marker gene analysis at different layers supported the hypothesis that different features are associated with various cell populations at different levels. Case study number 2. We applied DeepScena to analyze the 5,541 cells from 7 newly diagnosed or multiple myeloma patients. Our results demonstrate that the transformed latent space in DeepScena significantly improved the performance in separating potential cell clusters compared to the author's original labeling. So specifically, the original labels of the 10 cell types were divided into several small clusters, including CD14 monocytes, T cells, and natural killer cells. For example, CD14 monocytes were distributed across 3 groups, natural killer cells across 3 groups, and T cells across 4 groups. This indicates the possibility of further clustering these cell types into subpopulations. To explore these potential subpopulations, we applied DeepScena to the raw data, resulting in a total of 15 clusters. Our analysis not only reproduced the author's original annotations, but also effectively separated the CD14 monocytes, T cells, and natural killer cells into distinct clusters. Furthermore, the marker genes were detected and well support the different cell types, providing insight into the differentiation and activation states of immune cells and pathology. Alright, this is the basic introduction to DeepScena. Thank you for watching. If you have any questions or concerns, please contact me at sophiaroyuchen@gmail.com. Thank you.